You guys are all regulars. Well, good afternoon. This is Elik Esquilin. It's my pleasure to introduce him. Elik graduated with a BA in biotechnology from Miami-Dade College in the summer of 2025. While he was there, he conducted research with Dr. Tammy LaBerge on bacterial eDNA in polluted and non-polluted waterways of South Florida. With a lot of that research, he was able to present at different conferences and symposiums, including here at Archbold. And I had the pleasure of first meeting Elik when he came to the ranch to work on a collaboration using eDNA. And I was very impressed by his love of the outdoors, his friendly and kind personality, and his general enthusiasm. He really stood out to me. So when I heard that Elik was interested in one of our agroecology internships, he had my full support, and I was so excited that he was a part of our team. He's been an intern with us for about 7 months, and I can say that he has been a pleasure to work with. He's always willing to do any tough job we give him at the ranch. He's happy to learn and work really hard, and he's really contributed a lot to our team. And while we'll miss Elik at the ranch, he's not going far. On Monday, he's actually starting a field research position with the University of Georgia's Department of Genetics here on Monday at Archbold Biological Station. He is going to oversee a common garden experiment working with partridge peas. And so with that, I would like to thank Elik for being a meaningful part of our team and give him the floor to share his independent research project.
Hello, everyone. So my project is on the effects of nutrient addition and herbivory on Hypericum edisonium. Hypericum edisonium is a federally endangered flowering shrub that is only endemic to Lake Wales Ridge and the surrounding area. Particularly its effects on the plant's growth, reproduction, and long-term traits. So this project is in part with the Nutrient Network, also known as NutNet, which is a global long-term grassland study to measure the effects of fertilization on native grassland ecosystems. The project uses a standardized protocol with the same 3 nutrients, that being nitrogen, phosphorus, and potassium. And measures the long-term effects on plant biomass, species richness, and community composition. If you look on the map on the right, you'll see the over 200 active NutNet sites, for which Archbold's Buck Island Ranch is one of them. Fertilization. So as we all know, plants use nutrients to regulate their growth. It's more specifically, they use nitrogen to support vegetative growth and photosynthetic, chlorophyll production. They use phosphorus for energy transfer and reproductiveness, and then they use potassium to regulate water balance and stress levels. And actually, what a lot of NutNet studies have found is that long-term fertilization trends to increase plant biomass, but that's usually at the expense of species richness, which makes sense, right? What you're seeing is competitive dominance shift, right? You're seeing species that can more easily access this excess nutrients, outcompeting the species that can't. Which raises a question, right? How are native endangered plants like Hypericum edisonium affected? So like I mentioned earlier, edisonium is a federally endangered plant that is only endemic to Lake Wales Ridge and surrounding areas in subtropical grassland ecosystems. It actually depends pretty heavily on natural fire cycles and seasonal flooding to maintain its population. Unfortunately, as probably a lot of us know, Lake Wales Ridge has been developed and is increasingly being developed. A lot of that development being agricultural development, where nutrient addition and fertilization is pretty common practice. But we don't know the effects of fertilization on Hypericum edisonium, which is a pretty important question to ask when you're considering its conservation. So the way I measure these effects was by looking at plant traits, functional plant traits, like leaf area, specific leaf area, and leaf dry matter content, which kind of reflect how a plant acquires and uses and conserves resources. So leaf area indicates photosynthetic potential, which kind of makes sense, right? The larger the leaf, the more light it could absorb. Specific leaf area indicates acquisition of strategies, which kind of means like high SLA means thinner, faster growing leaves, where low SLA reflects thicker, slower growing leaves. And then leaf dry matter content indicates structural investment. So that's kind of like how tough and durable. So my project has 3 main objectives. Objective 1, determine how nutrient addition influences plant and reproductive traits of Hypericum edisonium. Objective 2, determine if herbivory influences plant and reproductive traits of Hypericum edisonium. And finally, objective 3, evaluate whether combined nutrient additions produce stronger effects than single nutrient treatment. Overall, my hypothesis is that nutrient addition will increase plant growth and reproductive performance, and these responses will differ depending on herbivore presence. Now into my methods. So the nut site on Buck Island Ranch is actually located within one of the semi-native pastures, which you can actually see its actual location on the ranch right there. These semi-native pastures have a history of no fertilization, very minimal alteration, and actually have a lot of native grass. Do have a history of fertilization, of management, of modification, have actually been seeded with more high forage grasses like Paspalum notatum, also known as Bahia grass, which is kind of well used in Florida cattle operations. Now the actual NutNet experimental design, the experiment uses the global NutNet protocol, which has the experiment separated into 3 separate blocks, that being block 1, block 2, and block 3, each block containing 10 plots. Each plot has an assigned nutrient treatment, of which there are 8 treatments. That being our control, our single nutrient treatments are nitrogen, phosphorus, and potassium, and then our combined nutrient treatments. Also within each block is a fenced-in enclosed plot, or 2 fenced-in enclosed plots, that being a secondary control and a secondary NPK. And this is to test nutrient and grazing interactions and excludes herbivory from wildlife like deer and rabbits. Whereas the entire NutNet project itself has a fence surrounding it to exclude cattle. So yes, it's an experiment at Buck Island Ranch, and we're not talking about cows. Thirdly, fertilization happens annually, and so does plant cover and data monitoring. For trait measurements, I went out in the month of November and counted flowers, buds, and fruits of every individual within a given plot. I also measured the height of the tallest plant and collected approximately 10 leaves from 3 individuals within a plot. I then scanned those leaves for leaf area, which you can see at the bottom down here, recorded fresh and dry mass, and then calculate specific leaf area and leaf dry matter content. Then for statistical analysis, I used RStudio to make statistical models to test the effects of nutrient and grazing treatments, effects on plant traits, reproductive count and plant height, and then finally specific level, species level cover over time. Now into my results. So this slide shows change in cover over time from 2015 to 2024. The top scatter plots being change in Hypericum edisonium, and the bottom being change in Paspalum notatum. Again, that is Bahia grass. That is the grass that's usually planted in improved pastures for cattle operations. The left scatter plots are our control, and the right is the combined nutrients of all 3 nutrients, NPK, nitrogen, phosphorus, potassium. Now, if we focus our attention to the top left, you'll see there's actually an increase in Hypericum edisonium over time, which is a good thing, right? That means there's a healthy population on our control that is actively growing. That blue line is our fence plots. Again, that's to exclude grazing from wildlife like deer and rabbits. And then the red line is only excluding cattle. Now, if we focus our attention to the right, you'll see there's actually a decline in Hypericum edisonium over time, cover over time, with more so predominant in our fence plots. So at first you might think that treatment itself is declining the population, but actually if you look down to the bottom right, you'll see that Paspalum notatum actually increases in cover over time, and more so in the fence plots. So actually what we're seeing here is what I mentioned earlier, and that's that shift in dominance. You're seeing Paspalum notatum be able to access that nutrients more so than Hypericum edisonium and outcompete it. And then this slide shows the treatment effects of specific leaf area on the left and leaf dry matter content on the right for all 8 nutrient treatments. Again, that's our control, nitrogen, phosphorus, potassium, and the combined nutrient treatment. And what we found actually is that specific leaf area and dry matter content really showed no significant response, which kind of suggests more of a conservative leaf construction strategy than compared to other plant traits. And this actually aligns with a lot of the literature that suggests that SLA and LDMC don't tend to respond to nutrient treatments as much as, let's say, other plant traits. Next is our treatment effect on leaf area. And what you might see already is that variation on leaf area across the plots is not directly tied to nutrient treatment. If you focus your attention on the control on the left, you'll see it's kind of a little bit higher than all the other nutrient treatments, which suggests it's right higher. But if we actually focus on the control and the NPK on the right and look more so at nutrient and exposure effects on LA, we'll see that there's actually a larger interaction happening between fertilization and herbivory. And what we're seeing is actually in our control treatments, the herbivory's effect is kind of insignificant. But then when we look at our nutrient treatment, you'll see it's quite significant, right? which is suggesting that fertilization across the board is increasing leaf area, but it's only apparent in our fenced fertilized plots because herbivores can't access and eat those leaves. This next slide shows the nutrient treatment's effect on reproductive structures and on the top and on height on the bottom, with control single nutrient treatments all combined into a single box plot, and then the combined nutrient treatments combined into a single box plot. And what you see is there's actually a increase in reproductive growth with the combined nutrient treatments and an increase in height with the combined nutrient treatments. Now into our discussion. So now if we kind of reflect back on our objectives, objective 1 was to determine how nutrient addition influences plant and reproductive traits of Hypericum edisonianum. And what we found is that fertilization did in fact increase leaf area, plant height, and reproductive structures, where SLA and LDMC kind of remained unchanged. Objective 2 was to determine if herbivory influenced these plant reproductive traits of Hypericum edisonianum. And what we found was in unfertilized plots, grazing kind of had little effect. But in fertilized plots, grazing reduced leaf area in the unfenced plots, and then fenced plots, full growth potential was expressed. Finally, in objective 3 was to evaluate whether combined nutrient additions produce stronger effects than single nutrient treatments, and the answer was yes, it did. So now if we look back at my hypothesis, it was largely supported. Nutrients did drive an increase in growth and reproduction, where grazing modified how those nutrient effects were expressed. Now to discuss all the findings. In unfertilized plots, there was limited growth and height, which suggests that plants allocated resources more conservable. Whereas in our fertilized plots, there was an increase in growth and reproductive potential supporting co-limitation. Also in these fertilized plots, we found that herbivory suppressed this nutrients and drove growth. Finally, we found that SLA and LDMC was not affected as much by treatment. For the future, I think continued long-term monitoring of Hypericum edisonianum is kind of super crucial for its conservation, continuing to investigate competitive interactions and nutrient enrichment influences. Also, I think it'd be quite interesting to look at how cattle grazing affects Hypericum edisonianum, because it is quite abundant in our semi-native pastures. Finally, I want to say thank you to LTAR for funding, to Archibald. Special thank you to Greg Sornier for helping me with and guiding me through the statistics of my project. And finally, thank you to everyone else. Questions?
I was wondering if cows really prefer this plant. I don't usually think of it as something super palatable.
So that's not anything we really know. From what I could tell, it doesn't look like it's grazed on the ranch. It looks like the cows kind of avoid it, to be honest. So, yeah.
2 questions. How do you, I'm sorry I missed this, but how do you measure reproductive potential? And second, yeah, like, did you ever find like caterpillars? Like, did you control for insect herbivory?
So we did not. There definitely were caterpillars, and that is not a part that we looked at. And what was that first question again? I'm sorry.
How did you measure reproductive potential?
So that was by counting flowers, buds, and
Oh, okay.
Yeah. Yes.
I was curious about the competitive interaction between the hair grass and the Hypericum. So do you think what's happening is just the hair grows so thickly that it suppresses germination of Hypericum, or what might be going on there?
So the truth is, it's probably more species than just the hay grass. It was just that was the clearest example to use. But yeah, it's probably more so like all these plants are covering it, taking over the nutrients that is in the soil for the plant. Yeah.
I'm not as familiar with the fire regimen over there. Is fire present, and like how would
it's actually avoided within Nunnet, but it is kind of present throughout the branch. They do fire things there.
Gotcha.
Yes.
Eric Mingus is online. He says, very nice study. As you know, Hypericum edisonianum is clonal.
Yep.
I'm wondering whether the changes in cover and leaf area that you found were more a function of larger ramets and or increased clonal recruitment or new stems and prints.
That's a great question. I think it'd be a great way to future explore the project. Yes.
Maybe an easy one. How wet is it out there? Are these in standing water on the ranch?
It definitely is seasonal. If it's the wet season, I think most of the time it's kind of flooded out in Nutnet, but for the past few months it's been quite dry.
Here it is.
For November, it was actually pretty dry. Not as dry as it is now, but yeah. There there wasn't standing water or anything like that.
A lot of the edisonianum populations here are kind of covered in these fungal galls, and that's part of our monitoring. I was just wondering if that's something you saw.
I haven't noticed it out there, so yeah, I have not noticed that at all. Thank you so much.